So I'm going to be talking about one of the most fundamental issues in biology, which underlies the existence of all living creatures and their ability to reproduce themselves. And whether we think of the very simplest of creatures, such as a bacterium, or something very much more complicated, such as ourselves, then one thing that binds us all together is that we're all built from tiny units called cells. And these were first observed in 1665 by Robert Hooke, looking down his microscope. And actually, this is a picture he drew of what he saw at the time, looking at a very thin section through a piece of cork. And what he saw is that the tissues divided up into these compartments that reminded him at the time of the rooms that monks live in a monastery, and so he called them cells. And it's now clear that all living things are built from these cells. And one of the most remarkable things about cells is that each individual cell has the ability to cut itself in two and make two equally viable living cells. And so one cell has the ability to grow and then cut itself in two, forming two cells. And if we're thinking of the simplest creatures like bacterium, then one cell is one creature, and so this forms two new creatures. Whereas with something very complicated like ourselves, we still start out as one individual cell, but after many, many, many rounds of this business of cell reproduction, we end up being able to make a body from very many trillions of cells that all cooperate together. So if we could look inside even the very simplest cell, the very simplest creature on Earth, such as this bacterium, it's a mycoplasma, you can see in this beautiful drawing, which is actually very, very realistic, drawn with incredible care and skill by David Goodsell, you can see that even in this very simplest of cell, there's an incredible sophistication of structure and complexity. And in fact, we couldn't draw an equivalent lifelike picture of the innards of a human cell because it's simply far, far, far too complicated. So this is just a very, very simplified, stylized cartoon, which is it's meant to illustrate that you could really argue that there is just as much complexity inside one human cell as there is in a whole human city, such as Dundee. And in fact, there's a number of analogies you could draw between them as well that really stand some argument. So just as a city needs an energy plant to produce all the energy that the city runs off, so too a cell has its own complicated structures that produce the energy that the cell needs for everything that goes on inside it. The cell has factories that produce all the building blocks that build absolutely everything in the cell. It even is able to export some of these to others, and there's a complex sorting system to deal with the export side of business. There's a very complicated transport network, defense systems, and so on and so on and so on. So to build all of this complexity requires inside the cell a vast amount of information, and that information is stored in a special compartment shown in red here in the chromosomes, which are very beautiful things because we can actually see them under the microscope sometimes. So this is a plant cell getting ready to cut itself in two and divide, and these worm-like things wriggling around are the chromosomes, which are lining up in the middle of the cell. And at some point, we're going to be able to see that each of what looks like one chromosome here is actually two identical chromosomes stuck together, which are then pulled apart beautifully, one of each pair to either pole of the cell. And when that is finished, then the cell cuts itself in two, as you can see indicated by this black line. So to survive this business of cutting itself in two, every cell has to do at least three key things. So the first is that it has to make one perfect copy of every one of these chromosomes so that, as we've seen, it can then separate each of these pairs of copies to either pole of the cell and then cut itself in two. And throughout my career and in my research lab up the road in the College of Life Sciences, we're interested in the first of these processes, how cells are able to copy their chromosomes. And it's an important process in biology because it underlies all of development of complex creatures. It's also one of the processes of biology that's very relevant for our understanding of human cancer, because it turns out that one of the very earliest things that happens in the development of a cancer is that cells acquire defects in this process of copying their chromosomes. So they pick up defects in the information that makes them what they are. And those defects drive the further development of cancer. And at least some mature cancers retain these defects in the process of copying chromosomes that then represents a kind of Achilles heel that can be exploited for cancer treatment. Because if you had a drug that interferes with this process of copying chromosomes, then that drug will be particularly bad for a cancer cell which has already got its own problems in that process. And that's really the challenge for cancer therapy, to try and find things that make cancer cells different from the rest of us, so that you could think of how to design a drug that might kill that part of you but without killing all of you. So it's important to understand how cells copy chromosomes, and to do that we have to understand what chromosomes are first and foremost. So every chromosome is built from one single very, very, very long molecule of DNA. And since the days of Watson and Crick in the early 50s, we've understood the structure of this DNA molecule, the double helical structure of this DNA molecule. Here's a model that Watson and Crick themselves built in 1953 that helped them understand this structure for the first time. But in fact, the structure of DNA is quite simple to understand conceptually, because it's really just like a zip that's been twisted. So if you take the DNA and untwist it, you see that you can then unzip it, so it's held together by teeth, just like a normal zip. And then you could zip it up again and twist it, and you're back to this double helix of DNA. So it's not that strange a molecule to understand. It's very much like a twisted zip. You just need to understand a couple of other features that are different to a normal zip. So the teeth holding the two strands of DNA together, in a normal zip, there'd just be one kind of tooth. In fact, in DNA, there are 4 different teeth that can hold these strands together, and we refer to them conventionally by the first letter of their chemical name, so A, T, G, and C, and then they like to stick together with each other, just like the teeth holding the zip together, only it turns out that A only likes to stick to T, and G only likes to stick to C. So by sticking together, these two strands build this molecule that, upon twisting, makes the double helix of DNA. And you can see that everywhere in this sequence, which you could read from one side to the other, everywhere you've got A on one strand, there's T on the other. Everywhere there's G on one side, there's C on the other, and so on. So the key point about DNA is that the linear sequence of these building blocks, these teeth of the zip of DNA, can be read from left to right as a sequence, and this sequence represents a code that contains all the information needed to build a living creature. And that's actually not such a complicated idea for us to understand, because we're actually really good at looking at complicated linear codes and understanding them very easily, much more complicated than DNA. So if you stare at this for just a few seconds, you'll see very quickly that this is a linear sequence built from a complicated code that actually has 26 letters in this case. And the first part of it seems to be nonsense, and then at a certain point it starts making sense. And you can see that then if you read each letter one after another from this point, it actually makes words that follow consecutively in a linear fashion, one after the other. And there's a little package of information here that at some point stops making sense. So in this linear sequence, we've got flanking sequences of nonsense with a package of information in the middle that we can understand. And in fact, that's very, very analogous to how the genetic code in DNA works, only it's much simpler. So the cell has to unwind these two DNA strands and then read through these letters of the DNA code. And it's simpler because actually all the words are 3 letters. So it reads through what is nonsense at first until it reaches a word that is A, T, G. And that tells the cell it's time to start paying attention, because from that point on, each 3-letter word in the DNA sequence corresponds uniquely to one of what are 20 building blocks of a different molecule, which is a protein. So the DNA code tells the cell how to make proteins, and the proteins are all the building blocks that make the cell what they are and do all these fascinating tasks that are going on inside the cell. So the linear sequence of the DNA corresponds to a linear sequence of one protein, and at the end a word TGA tells the cell to stop so that it's just made this length of protein. Now each DNA molecule, one chromosome has one DNA molecule. It's really, really long, and it then contains many genes that have the code to build many, many, many proteins. So I've said that each protein, a bit like DNA, is a linear sequence built from one subunit stuck to the next. And there are 20 different ones in proteins, but only 4 in DNA. But there's one key difference, because in the DNA, whatever the sequence is, the structure is always going to be that beautiful double helix that I showed you. But the key thing about proteins is whatever the linear sequence of the 20 building blocks is as a protein makes it then spontaneously jump into a unique, different structure. So every one of the 1,000 proteins has a different structure in the cell, and that's why these proteins can do thousands of different things. They can be building blocks, the bricks and mortar, they can be machines that make things happen. So these are actually the structures of 4 real proteins, and you could take any one of these and pull it apart if you could, if your hands were small enough, and you'd see that you have one linear chain built from 20 building blocks, but the unique sequence makes it adopt this structure and not that one. So the cell contains in its DNA the information to make these thousands of proteins that make the cell what they are. So the question we're interested in is, how do cells copy the DNA and so the chromosomes? And so how do they copy and are able to inherit this information from one generation to the next? And the answers actually lie, above all, in the DNA itself. So Watson and Crick noticed back in 1953 that the structure, the double helical structure of the DNA, suggested straight away the basic principles of how you copy DNA. What the cell has to do is take a double helix, unwind the 2 strands, and then use each one as a template to build the opposite, remembering that principle of A always sticking to T and G always sticking to C. So you'd be able to take this strand and bit by bit build the complementary stand from what I've told you, just putting A, G, C, and T where they should be. So our question is, how does the cell actually do that? So what's the molecular machinery that lets a cell copy a DNA molecule and so copy its chromosome? And the answer then, of course, is by using proteins, because proteins do all the jobs in the cell. So there are 2 key proteins in particular involved in this process, one of which is actually it forms a ring around one of the strands of DNA and slides along actively, displacing the other strand. So this is the thing that unwinds the 2 strands, and because it unwinds a double helix, we call it a DNA helicase. And then another protein that is able to take these unwound strands and now polymerize a new DNA molecule, putting ATGC in the right positions to build a new DNA chain. And because it does that, we call it a DNA polymerase. So we actually only understand the machinery that does this process in one species at this point of all of life on Earth in any great detail, and it's a bacterium. And the key thing we've learnt, really, is that the DNA helicase, which is the blue thing here that's unwinding this double helix, and the polymerases, the purple things there, they actually don't work on their own, but they're built together with lots of other factors that we don't need to worry about today into a larger machine, and the machine is needed to do this job, so to copy the DNA that makes a bacterial chromosome. Now, I've told you that every chromosome is one molecule of DNA, and that's true whether we're talking about bacteria or a human chromosome here, but a human chromosome's different, quite different, in that it's very, very, very much longer than a bacterial chromosome, and also the DNA is wrapped up with very many proteins that control its function. So it's not hard to believe that copying one of these is more complicated than copying one of these, and in fact it turns out to be very much more complicated. The machine's much bigger, there are many more components, and we still have lots of unanswered questions to try and understand this. So the position of our understanding at the minute is rather like if we'd given someone a disassembled car and asked them to rebuild it. They'd know that they've got all the bits and pieces there in front of them, and they'd know what they're trying to do. They're trying to build something to take you very fast down a road, and they'd understand some of the bits and pieces. So they'd recognize the chassis, and I know what a seat is, I know what wheels are. And then I hit a few problems where there are lots of things here that I know they fit somewhere, but what they do and why did they need to be there in the first place, and what was the point? And that's roughly where we are with our understanding of this machinery in human cells at the minute. So here's a few questions that we're struggling with for now and the future. So we'd love to understand in great detail the mechanisms that underlie that machine. So why does it exist? Why can't the bits and pieces work on their own? What does each bit do in order to understand the whole? And then one of the most remarkable things about this machine is that copying one of those enormous human chromosomes with all that DNA sequence that makes us what we are, in many ways, the cell is able to do it, the machine is able to do it without making almost any mistakes. It's a phenomenally successful process, and that's really important because mistakes in the DNA can be lethal, they could kill us, or they can lead to genetic diseases such as cancer. So how does this machine manage to be so good is a very complicated question that is very important for us to understand. And then ultimately we'd like to understand how does this process, how does this machine actually go wrong? Why is its action impaired in the early stages of cancer development? So that ultimately we could try and think which bits of this machine, as we get to know it better, might be the best bits to target with future drugs that could be tried out in cancer therapies. So on that note, I'll finish, and thanks very much for your attention.